I don't know the names. That's like Mander. Char, yeah, my city and my thing. Yeah, Charmander. I know that one. I know Pikachu.
I like adding juice.
Well, that has to be something. It's for applications. And so how do we go from this little cute thing to, well, it's still cute, I guess, but it's bigger, it's stronger, it's whatever. What's that one called? Nice. Yeah. Jaman, get some points on the board. We are so sparsely populated today. I guess, yeah. So we're going to start getting into applications. We'll finish up AMPK. This will be the last time we talk about AMPK. The whole mTOR map will be mapped at the end of today. We'll have talked about everything on the mTOR map. And from the second half of today and into Monday, it's just going to be how do we use that map? How do we navigate that map to elicit outcomes that we want? Now, whether that's to inhibit mTOR, and if it's cancer, if it's diabetes, something like this, where AMPK is advantageous, then how do we do that? But mostly I'll be talking about how to activate mTOR for athleticism, for hypertrophy, for growth, right? To go from the little fire guy to the bigger fire guy. And what kind of juice is gonna work. So apple juice, sure, you'll get an insulin response. You get an insulin response. Insulin, you'll get PI3K, PKB signaling and get some mTOR. But with insulin, you tend to deposit more fat than muscle. Bodybuilders, semi-notoriously use insulin as one of their anabolic agents. And it is anabolic, but man, it's anabolic for fat. It's not as anabolic for muscle. It is, but there are things that work so much better than insulin. And so we'll talk about some of that stuff. The points are updated, you know where to find stuff. You know the chemicals, right? We have a bunch of cell signaling cascades, mostly PI3K, PKB. There's overlap with MAPK. Mechanical tension, you know we have all these mechanosensors. We have these proteins that sense loads and transmit that information in the form of chemical signals into the cell. But you can bypass PI3K, PKB, you can bypass that Wortmannin inhibition, you can bypass that. Endocrine system, again, is mostly cell surface receptors. If you're a steroid hormone, you just wiggle your way in. You're made from cholesterol, you can get through a phospholipid bilayer. For nutrition, carbs, fats, and protein all matter. And proteins are really what matter the most because amino acids are the way that you get mTOR to the lysosome, mostly to the lysosome, that's its primary work site. Other areas in the cell, mTOR can work, but the lysosome is the primary one. And getting that RAG-A GTP loaded, that's what amino acids are doing. And you need that GTP loaded RAG to have mTOR be functional, be able to be activated by Reb. If Reb is off, is not GTP loaded, or if the RAGs are not GTP loaded, mTOR is not on. mTOR will not be on. You need both of these things. And proteins, amino acids, in particular, like leucine and arginine, methionine, lysine works, glutamine works. Certain amino acids are much more effective than the others. The two most important ones would be leucine and arginine for the stimulation. But on Monday, we'll talk about restriction, about protein restriction. And methionine is an important one for protein restriction. If you want to do fasting, protein fasting, stimulate autophagy without actually eliminating all of your calories, all of your protein, the methionine is an important one for that.
So is it just RAG-A that's GTP loaded? Or,
yeah, but just, you don't need, in your drawings, you don't need to say that. You can just say RAG as a whole thing. RAG, and just put a little GTP on it.
Because RAG-A and RAG-B are together, they're not.
They're together, right? But just put RAG and a little GTP on it, and that's sufficient. Technically, RAG-A is the GTP-loaded one when mTOR is on. And so proteins, that's really what the amino acids are doing, are relocalizing mTOR, that complex, to the lysosomal surface where it can do its kinase activity. Now, Reb permits its kinase activity, but it can't have its kinase turned on if amino acids haven't put it within proximity of Reb. The fats, the carbohydrates, these are more about Reb activity than RAG. Fats and carbs are more about Reb than RAG. So one more time of reading this, the phrasing is a little bit different, but what turns on, big picture, mTOR, what turns on mTOR? Number one is need, right? Need is really what does it. So subjecting tissues to loads causes structural damage with a degree of damage corresponding to the magnitude of the load. That damage itself releases chemicals. Damaged tissues release chemicals. Remember those myokines that get released from the muscle, 600 of these things, all these little proteins being released from the muscle itself. Damaged tissue releases chemicals. More damage, more chemicals. Those chemicals notify the cellular machinery that repair is in order. The mechanical load informs the cell of what the current stress is. Whatever it's undergoing in this moment, the mechanical load is informing the cell of those stresses. And the inflammation informs it what its recent stress was. What did that cell just undergo? That's what the chemical signals are informing the cell. Here's what you need to fix now, and here's an estimation of what's coming up. I don't have any resources to offer you. I'm just letting you know how to prioritize. So need is the reason mTOR is responding. Those mechanical sensors are sensing what the cell is currently doing. You know, if the wind is blowing really hard against a house, I'm gonna have to do something about this in the morning, right? The tree branches are falling off. I might have to fix my roof in the morning. And then the next day, when you're looking at the house and you see like holes in the roof and stuff from these fallen branches, okay, that's sort of the chemical signaling of the damage. Need, you need to repair things, but need isn't providing the nutrition. Nutrition is a separate signal. All fauna fast and they feed, and their bodies need a way to detect which state they're in, if they're fasting, if they're between meals, if they're denied sustenance, or if they're currently indulging. So the bodies need a way to detect which state they're in, fasting or feeding, so it can respond appropriately. It would be dangerous, at least dangerous, if not lethal, certainly wasteful, to behave in any other way. To go to great expense when you don't have the income, right? You need to be sort of cheap when money is scarce, and you can be a little bit more extravagant in your spending when money is abundant, when it's plentiful. So that's the nutrition part. The body recognizes, is there a surplus or is there a deficit, and it budgets accordingly. And then the assumption of nutrition. So I put some flavor into this one. Consider the life choices of an unemployed person named Jake the Hog Catcher Matterhorn Jr. If Jake THCM Jr. is responsible, he'll spend his money sparingly. He doesn't know how long his savings will need to last, but if he's informed of a big windfall headed his way, the reluctance to spend his savings loosens its grip. The same thing happens when your cells anticipate the arrival of caloric income. An injection of insulin notifies them that they've just won the Publishers Clearinghouse sweepstakes, or at least a biological equivalent of that. The money hasn't yet arrived, but go ahead and translate a new car. So there are ways to trick your body, and you can do something similar if you do like a carbohydrate rinse in your mouth. Don't actually eat the carbohydrates. Put some carbs in your mouth and spit it out and rinse it out. And will you absorb like the tiniest bit? Sure, but the tiniest bit, that's not enough to offset nutrient depletion-induced fatigue. but the signal of carbs in your mouth makes you perform better. If you're doing an endurance activity, you can see power output increase without even getting the carbs. You just put carbs in your mouth and then spit them. And you will perform better because your body thinks those carbs are coming. So there's some physiological trickery that you can employ. And as we start getting into some of these applications, a little bit can be physiological trickery. A little bit can be, let's manipulate the body's assumptions to elicit the outcome that we want. So AMPK, probably the most catabolic thing in the body, the most catabolic enzyme in the body. And remember, enzymes do all of the labor. A hormone, if you take an androgen receptor blocker, testosterone in circulation will go up, but testosterone isn't doing anything. These things have to bind. These hormones, it's growth hormone, it's IGF, it's insulin, it's testosterone, whatever it is, these hormones have to bind to the receptor to have any effect at all. Insulin in the blood, testosterone in the blood, that's not really helpful. That's not doing anything. And you can increase the amount of these hormones just by making it so they don't bind. When they bind, when these hormones bind, certainly when polypeptide hormones bind, protein, amine, polypeptide hormones, when those bind, it's a bunch of enzymes doing all of the work. And there are other ways to interact with those enzymes. Hormones, that's one way. And it's a really cheap way of talking about physiology. It's like, oh, hormones do this effect. Not really. Hormones initiate enzymatic activity that does all of that effect. And there's other ways of having that interaction. So AMPK, really important enzyme. And on this side, we see an interaction with tuberin, phosphorylating tuberin, turning tuberin on, so that tuberin is preventing red from activating mTOR. So AMPK is blocking mTOR at the tuberin step. AMPK is also phosphorylating raptor and turning it off. So raptor is unable to help recruit these downstream targets for the mTOR kinase, for the enzyme to do its phosphorylating. So AMPK, and then there's also evidence that AMPK phosphorylates mTOR directly. And so there are multiple modes of inhibition for AMPK to shut off anabolism, to shut off protein synthesis. And part of that is just repair, it's basic repair, right? Go exercise and just get back to baseline. Let's just put this stuff back together. This is included in that. Now you're not going to, if you, let's say you're on, you're taking Wortmenin or something, you'll still be able to translate some proteins. You don't just abolish all translation until you wither into nothing. But this is a huge impairment in both recovery and especially hypertrophy, taking a tissue and making it bigger and stronger, reinforcing it with additional proteins above and beyond what existed before. And then on this side, if you turn AMPK on, you're turning on protein degradation. Great for some things, great for some goals, terrible for other goals. If you are a football player, if you are most athletes other than endurance athletes. Endurance athletes want AMPK because you're getting things like mitochondrial biogenesis, you're becoming a better endurance athlete in the presence of AMPK. But most athletes, with that exception, do not want AMPK on, they want it off. So this is just a look at what it's phosphorylating on the mTOR side, on the protein translation inhibition side, blocking the synthesis, the translation, the synthesis of new proteins. So again, raptor, tuberin, and mTOR directly, probably the two most important ones, though, are that raptor and the tuberin, the bold ones up there. And just remember, I refer to it as St. Peter because it really is the metabolic decider of whether you're currently anabolic or you are catabolic. AMPK is the decider of that. And the way that, and it does other stuff too. It's going to do other things too. The way it makes its decision is the ATP to AMP ratio, and ADP in there as well. ADP matters too. But it's going to regulate carbohydrate, right? So it will block glycogen synthase. AMPK will block glycogen synthase. So instead of taking sugar and linking them together and storing it, it's going to facilitate the breakdown of carbohydrate. Hexokinase, PFK, GLUT4 translocation. Let's get carbs out of the bloodstream, just like insulin would. Let's get carbs out of the bloodstream. But instead of turning on glycogen synthase, which is what insulin would do, let's break those carbs down and immediately generate ATP out of them. Also, mitochondrial biogenesis, let's get long-term aerobic capacity, because energy stress is really what turns on AMPKs, energy stress. Energy stress is that ratio of ATP to the other ones. ATP to ADP and to AMP, that's energy stress. And how might you resolve this in the future? Think specificity of adaptation. The stress that your cells and tissues, the stress that your body is under is one of energy depletion. What's a good way to resolve that in future bouts of that same exposure? Let's get some more mitochondria in there. Let's become more oxidative. Let's build our ability to use oxidative metabolism. And then it also promotes fatty acid oxidation too. And as we said on the previous slide, protein degradation and inhibition of protein anabolism, just like it's inhibiting anabolism of fat and anabolism of carbohydrate. It's catabolism of all three macronutrients, carbs, fats, and proteins. Now over here, there are multiple subunits, right? So here's your AMPK, it's a kinase, it's gonna phosphorylate a ton of stuff. Over here on the gamma subunit, that's where the ATP and AMP and ADP bind. And if you have AMP bound to the gamma subunit, that permits upstream activation. Liver kinase B1, tons of literature in the liver. It behaves really similarly in the liver and in skeletal muscle. The primary promoter of AMPK in the skeletal muscle, though, LKB1, and that's at the alpha subunit. That's what phosphorylates over here, turning on AMPK. But AMPK, it also has to recognize carbohydrate levels, right? Because if it is blocking glycogen synthase and turning on glycolysis and taking carbohydrates into the cell, it has to know what your carbohydrate levels are too, not just the levels of ATP, ADP, and AMP. It has to understand how much carbohydrate you have on board if it's going to be cranking up carbohydrate metabolism. So glycogen binds to the beta subunit. If you have glycogen in abundance, glycogen binds to the beta subunit, that's inhibitory. Now the way to shut AMPK off is a phosphatase, right? So you have a kinase is going to turn it on, we're going to phosphorylate that subunit. We have to dephosphorylate it to shut it off. If ATP binds over here, that permits its dephosphorylation by a phosphatase. If ADP or AMP are bound, you're good luck dephosphorylating that thing. So once you resolve the energy stress, once you resolve your ratio of ATP to ADP and AMP, and you generate a lot of ATP and you buffer those other two, that's when ATP will bind, and you don't really need AMPK active anymore because you've already resolved the energy stress. So at that point, you can dephosphorylate it.
Glycogen's dephosphorylating?
Glycogen is inhibitory to its activity. Yeah.
Okay.
Yeah. So glycogen is a inhibitory agent for it. It's not, if you have an abundance of glycogen and just nothing but AMP, it's something to be on. This isn't as powerful as this. Kind of like when we were talking about, like caffeine versus insulin. And insulin is, if we're going to go through PDE, phosphodiesterase, insulin is a really potent activator of phosphodiesterase, and methylxanthines are a weak inhibitor. And so there's a potency and a weakness This is really potent over here. Just another look at everything we were just talking about. You're exercising, whatever you're doing. If you are consuming a bunch of ATP, that means you're producing a bunch of ADP and AMP. At first, ATP hydrolysis, right? You take your ATP, chop it up, you get your ADP, and phosphate and hydrogen ion, you get your ADP. And then the adenylyl kinase reaction, you take a couple of those ADPs and convert them into an ATP and an AMP. Now we are consuming the ADP as well. So it's ATP and ADP, but we're still building up a bunch of ADP. ATP levels, how much this goes down depends on the nature of exercise. Originally it was thought the ATP levels in a tissue, like, oh, these aren't going down below about 80% of their resting values. That's what was said initially, that these cells, oh, it'll go down about 20% to about 80%. And then people started evaluating cell by cell activity and found at the whole tissue level, there are a bunch of cells that aren't active. Those cells are resting, and that sort of mean value distorts our perception of how much ATP gets depleted. In an individual muscle cell, you might get down to about 20% of your resting ATP. You might go down by 80%, not to 80%. The preposition changes, and it makes all the difference in terms of how much ATP depletion we have. But still, ADP and AMP, that change is astronomical compared to the amount that ATP changes. The ATP change, even going down to a fifth of its resting value, is still nothing compared to the change of ADP, which is nothing compared to the change of AMP. So that AMP really builds up and binds to that gamma subunit. The AMP binds there. Now, if AMP or ADP is bound, you're not going to dephosphorylate this thing. You're going to keep this thing turned on. But AMP is really how we're activating this. And in skeletal muscle, LKB1 is this primary upstream kinase that's going to do the activation. And remember, that's where the carbohydrates are going to bind. So ATP hydrolysis, adenylyl kinase, build up a bunch of AMP. AMP turns on AMPK, adenosine monophosphate activated protein kinase, AMPK. This is just a narrative version of the carbohydrate stuff. Now there's a ton, in last, what is Friday, on Wednesday's lecture, oh, there must have been like 30 PDFs in that slideshow. And we didn't actually get to all of them in the slides. So if there are some PDFs that appear in this one that aren't in this file folder, if you check, if you're interested in looking at these PDFs, you don't have to. If you're interested in looking at them, just look at Wednesdays. If you see one of these, the title of an article, and you check today's file folder and you don't see the article in that one, just go back. There's like 30 in that one. But this is the one talking about carbohydrates, and AMPK has to sense the amount of cellular carbohydrate as well, since it is regulating carbohydrate metabolism. So again, you've seen this slide before, ATP hydrolysis. Chop up a bunch of ATP. We're gonna get a bunch of ADP. Adenylyl kinase. Let's build us some AMP. We're now accumulating AMP. Normally, we have AMP deaminase, and normally I rely on Cameron to explain to me the drug reaction that we would inhibit that. But ATP and AMP are competing for that gamma subunit on the AMPK, and depending on that ratio, when you really build up AMPK, just as a matter of concentration, the one that is more concentrated is going to bind. If you were to, let's say there's like a jar and it's filled with 1,000 Skittles and 1 M&M, and you reach in at random and pull one thing out, what are you pulling out? Unless you're like super lucky, you're pulling out Skittles. Yeah, if there's 1 M&M and 1,000 Skittles, these concentrations determine what binds. And so when you change concentrations, that determines the biochemistry that follows. So with AMP bound, the upstream kinase LKB1 can activate AMPK. If AMP or ADP is bound, you can't dephosphorylate it. It remains on. And if it is on, if AMPK is on, you remain catabolic. You remain unable to carry out hypertrophy, at least effectively. You're going to have a really hard time regenerating tissue and hypertrophying protein accretion, accumulating protein. Exercise, hypoxia, exercise during hypoxia, glucose starvation, carbohydrate starvation, any type of prolonged fasting, all of these things are going to activate AMPK. All of these things lead to energy stress. Go up the mountain, be short on breath, short on oxygen, have the partial pressure of oxygen get low, be short on breath. And you will activate AMPK because that's energy stress. You're not getting as much oxidative metabolism, that's energy stress. AMPK is going to be turned on. So all of these things, you are going to change the ratio of ATP and AMP. Now, reactive oxygen species also matter. There's a number of places where reactive oxygen species play in as a regulator. But remember, reactive oxygen species are a regulator of a lot of things. They're a regulator of mTOR. Reactive oxygen species have a role. You don't need to know that role. From what I've read in the literature, much of it remains cloudy in its balance between anabolism and catabolism. During exercise, an excess of reactive oxygen species seems to be more catabolic. Physiological reactive oxygen species levels corresponding to resistance training, what you would normally expect in reactive oxygen species, does seem to be an anabolic signal. Okay, so we ended here on this slide on Wednesday. And you know all of this stuff. We talked very briefly about these three. We'll talk a little more detail about them. But there's LKB1 activating AMPK, is the alpha subunit. The gamma subunit over there, right? That's where the AMP or ATP or ADP is going to bind. The beta subunit, that's where the carbohydrates are going to bind. There's PKB, phosphatase intensin homolog. This is converting PIP3 into PIP2. PI3K, phosphatidylinositol 3 kinase is kinasing, it's phosphorylating PIP2 to make it PIP3, so that PDK can activate PKB. So there's PKB, there's the tuberin, the tuberous sclerosis complex, TSC2 is tuberin. There's RED, mTOR, Raptor, PRAS40, the downstream targets. You know all this stuff. Rapamycin over here, which would bind at the FKBP12 site. There's the IGF-1 receptor. Now, insulin could be initiating this. Some myokines could be initiating this. In this case, we have some sort of growth factor. It's IGF-1 here. Now, for this, though, to activate AMPK, there's ICAR, AICAR, Metformin, resveratrol. We'll go through these individually, but these are just three examples of drugs or polyphenols in the case of resveratrol that will interact with AMPK, that will turn on, promote AMPK. So resveratrol is, again, it's a polyphenol. It's in your red wine, it's in grapes, and it promotes LKB1's activity. In higher doses, it's going to change the ratio of increasing AMP, change the ratio between AMP to ATP. But resveratrol is what people say makes red wine healthy. Oh, it has resveratrol. Okay, just eat like 2 walnuts, and there's your bottle of wine. I don't know the exact numbers, but you don't need to eat much of some of these foods, and you have like your whole bottle of wine worth of resveratrol. So drinking to excess because it's healthful, resveratrol might not be your go-to compound, but it does promote LKB1. This is action, promotion of AMPK. So resveratrol does promote AMPK activity. And at the end today, we'll talk about AMPK's good parts, lifespan, heart health, things like that. Metformin, now I keep saying these drugs work in a number of ways. All of these drugs have a number of interactions. One of the interactions for metformin is blocking AMP deaminase. Remember, you build up AMP and you have to eliminate that AMP pool, AMP deaminase. So let's convert it to IMP, inosine monophosphate, and ammonia. You dispose of that AMP. But if you don't dispose of it, AMP levels build up. Metformin has additional effects. Tons of effects. Not all of them are characterized. My hunch is not all of them will ever be characterized. But it also, if you look at the electron transport chain, the first complex in the electron transport chain, there's inhibition there as well. Lots of things are going to interact at that first, where NADH does its delivery in the electron transport chain. FADH2, that will still work just fine. Remember, cyanide is what's going to obstruct cytochrome C oxidase, and that's terrible. That means like you're dying with cyanide. These things that obstruct the first complex, that's not bad. That's just your cells have to sing a little harder for their supper. You're going to change the ATP to AMP ratio when you exert a little bit of inhibition at that first one. So metformin, that's a secondary effect of metformin. Also, a lot of these drugs and polyphenols and compounds that enter on AMPK. A broad range of targets. Adiponectin release from fat cells. That will help turn on AMPK, and sometimes these will increase adiponectin. So there's a lot of effects of these things. Now, the ICAR, I keep talking about, AICAR, banned in cycling. Right, these are 2019. There's a New York Times article, doping cloud still looms over a thrilling Tour de France. They're not, EPO is boring. Does EPO work way better than this? Yeah, but everybody knows what it is, and everybody, EPO has been around long enough that nobody's excited by it. Introduce something new. New is always exciting. New doesn't mean better. In 100 years, what's on the radio today compared to like the Beatles or Bach or whatever? I don't know that people are going to look back 100 years later, but no, those are just as good. New doesn't mean better. New just means exciting. Whatever is on TV now, like you ask people what their favorite show is, half the time it's Friends or The Office or something. Those aren't new. But people would rather watch what's new than old reruns of Friends or something. So new doesn't mean better. New just means we're paying attention to it, it's worth writing about. And when you introduce this to cycling, they do perform better. How does it work? It's an AMP mimetic. It mimics AMP. So it binds to AMPK. So it facilitates the action of AMPK. So it facilitates aerobic metabolism very effectively. This article goes through most of the known compounds that are going to interact with AMPK. So indirect activators, metformin, there's one of the indirect promoters of AMPK. There's resveratrol right there. It's just sort of little scribbles and gibberish and stuff. It's hard to read these things. And I don't know. Let's see. That berberine. You remember berberine? We talked about that one as a generally effective fat loss drug. I think it was on the it was like the #1 compound of the maybe list was berberine, promoter of AMPK. And curcumin is on the list over there, another promoter of AMPK. So there's a lot of natural, like again, these polyphenols. Just eat a bunch of colorful food. Go eat a big salad. Eat a salad with some berries and nuts and stuff on it, and you're going to get a lot of these polyphenols. And these polyphenols, many of them are going to promote AMPK, just like metformin would. And, a lot of these are diabetes drugs. And again, multiple targets for these things. But many of these things, lots of the drugs and polyphenols, not all of them, but a lot of them are inhibiting the first complex in the electron transport chain. And if you do that, the electron transport chain still works. If you inhibit cytochrome C oxidase, goodbye electron transport chain. That's critical to survival. If you get in the way of complex 1, if you put some speed bumps on the complex 1 road, you're going to change the ratio of ATP to AMP. You're going to activate AMPK. It's going to sense some modest energy stress, and especially in the presence of exercise. But there's also direct AMPK activators. And you notice the first one is AICAR, a direct activator, meaning it actually binds to AMPK. It's not just changing ratios of things. It binds, it's a mimetic, it mimics AMP and binds to AMPK and is a really strong promoter of AMPK. Again, if you're a cyclist or an endurance runner, that's really helpful. Get all this AMPK.
The gamma subunit?
Yes, to the gamma subunit. Yeah, it binds as though it's AMP. Yeah, to the gamma subunit. And then just, some other kind of drugs. Now, what is the reason that AMPK knocks out protein synthesis? Why does AMPK care about how much protein you're translating, about protein accretion? It is so expensive. It could be half of a cell's energy expenditure is translating proteins. A full 1/3 to 50% of that cell's entire budget, its entire energy expense, is protein translation. If you're trying to save money, if you're trying to reconstitute ATP, if you're trying to stop spending ATP and start accumulating it, look at the other side of the equation, not just how much money you're earning, but how much are you spending? This is like when somebody's trying to balance their budget. And they're like, well, I'm going to work 10 extra hours this week. I'm going to sell some stuff on eBay. But they don't offset their gambling, addiction. And like, okay, well, you're losing 10 grand a month through gambling debt. Yeah, but I put in, 10 extra hours this week and it was overtime. You're gambling away $10,000 a month. That's protein anabolism. It is so expensive. And so again, if you want to have an effective weight loss program, translation is pretty effective. If you want to lose weight, go translate as much as you can, and there's a metabolic price tag to that. So AMPK is going to cut out those major expenses. mTOR, super expensive, which is the reason if you have GTP-loaded rag forever, you're not going to last long. There's rodent models of that. They don't last long. They just translate themselves to death. Regulation is key, and regulating expenses, that's what AMPK is doing. Now, in terms of what it does, what AMPK is going to do to skeletal muscle, these are mouse models. But lacking AMPK. Mouse models in which they lack AMPK, so lacking over here, not lacking here. Look at the size difference in these muscles. We only get 50% or so larger muscle mass in the rodents who are lacking AMPK. The inhibition of growth. People will talk about myostatin, this inhibitor of growth, and you see those Belgian blue cows. AMPK, very similar. If you can get rid of AMPK, the growth is increased a great deal. And this is just narrative from the same study. Muscle cells lacking P70S6K, immediately downstream from mTOR. Muscle cells lacking that exhibited reduced cell size and failed to adapt their size to the inhibitory effect of rapamycin amino starvation. Suppression of P70S6K signaling triggers an energy stress response defined by an increase in the AMP-ATP ratio. But it goes on to say, in contrast, loss of AMPK induces an absence of P70S6K inhibition and leads to a higher protein synthesis rate and ultimately increased muscle cell size. So AMPK, the longer that's on, the more inhibition of mTOR you have, the more inhibition of growth that you're going to have. And again, same study, you should recognize those things, right? So treatment of rat cardiomyocytes, now they're looking at heart cells as opposed to over here we're in a soleus. With cardiomyocytes, with metformin, icar, resveratrol, activated AMPK and inhibited the development of hypertrophy. Turn on AMPK, turn off hypertrophy. And here's another study that was looking at mechanical loading signaling. And what is the hypertrophy response, the hypertrophic response to mechanical loading and loss of AMPK versus wild type. This is phosphorylation of P70SSK, phosphorylation of ribosomal protein S6. There's day 0, day 7, day 21. It's not like, well, it'll get back to baseline. We'll figure things out. We'll get back to baseline. No, day 7, day 21, and we're seeing a massive difference in the amount of that immediate downstream target from mTOR, P70SSK. The amount of that that's phosphorylated, meaning the amount of mTOR activity that's currently happening at day 21 is way bigger than day 7, certainly day 0. And ribosomal protein S6, we're seeing the same thing. What does that translate to in terms of hypertrophy? This is percent hypertrophy, what you would expect, right? There's AMPK, absent AMPK, and then here is wild type. The less AMPK activity that you have, the bigger your muscles grow. And you can induce atrophy by promoting AMPK. FOXO. Remember when we're looking at these ULK1 and FOXO and protein degradation signals that AMPK transmits? We're seeing this increase in FOXO signaling. What does that translate to for the protein content? It's just going down, 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 down. So AMPK blocks hypertrophy and initiates atrophy. Great for cyclists, terrible for anybody trying to do non-endurance things.
So what if, like I say, you just ran a marathon, you have extreme levels of AMPK, and then you just consume a lot of protein after? Does it kind of cancel out or not?
We'll talk about that the exact question on Monday. I'll get into detail. The very short answer is, it depends how trained you are. It depends how far along in a training program you are. And it's a really important question, this balance. I'm going to spend like half an hour on this question on Monday. But balancing anabolism and catabolism, and a lot of the proteins you have to translate correspond to the activity that you're doing. And so you will translate proteins corresponding to the stressor. It's not like you're going to get big, running a marathon, but you do have to translate a bunch of proteins according to that imposed stress. And depending on how early this training is, if you're running a marathon, you've probably trained for quite a while. Early on in training, you can really do everything. Your body just responds abundantly. You can do some anabolism and you can improve in endurance and everything early on in training your people. And then the body starts to develop a phenotype. And then they oppose each other, especially endurance is going to oppose anabolism. The more trained you get, the more you establish your phenotype, training-induced phenotype. These things start to compete with each other a little bit. But there's always ways to interact. How much feeding can you do? Can you get some IGF going, and can you get a bunch of protein on board? It's in particular the right amino acids. Now there's another thing I'll show you. It's Dave Hooper's research where endurance exercise depletes testosterone. And so that secondary signaling of testosterone, that would be a second challenge to overcome in the presence of long duration endurance activity, trying to combine that with anabolic signaling. You have to do it with a little bit less testosterone, and aerobic stress presents challenges. We'll talk in detail on Monday. I promise I'll do like a 30-minute answer to that question. And I'll show you the studies. Now, in the hypothalamus, we know the 6 Fs, right? One of our teams is one of the 6 Fs. One of those F's in what the hypothalamus is regulating is feeding. Food, feeding, appetite. And so stuff is going to affect the hypothalamus. Ampk is one of those things. Ampk is going to affect the hypothalamus. Now, insulin and leptin, we're seeing similar signaling in the hypothalamus, right? Leptin, satiety hormone. All right, we don't need to keep eating. You know, we don't need to be all ravenous in the moment. We have a bunch of insulin in our system. We obviously just ate a bunch of food, a bunch of carbs probably. And so we don't need to continue eating. It's okay to feel satisfied. Satiety is a reasonable sensation in the moment. So insulin, leptin, satiety, that's fine. So that's over here. Now, food intake. Remember, hypothalamus regulates a bunch of stuff. And food intake is one of them. Leptin is inhibiting AMPK in the hypothalamus. That's exerting its effect, AMPK in the hypothalamus. Now remember, AMPK, it's trying to manage your energy stress. One way to manage that is put food in your mouth. Now this is a longer term solution, breaking down the carbs that are in your bloodstream right in this second, get them into the cell and break them down. That's a faster resolution. We gotta replace those with something. So AMPK in the hypothalamus is going to initiate feeding. It's going to stimulate hunger, appetite. Cannabinoids, go smoke your weed. You get the munchies. What is that doing? Stimulating AMPK in the hypothalamus. So you get the munchies. That's what, you know, THC induced like feasts. You just, oh, I just can't, you know, get enough food in my mouth. How many boxes of crackers did you go through today? That's cannabinoids. That's the effect of increasing AMPK signaling in the hypothalamus. Ghrelin, the opposite of leptin. Ghrelin, this is your food-seeking hormone. Remember, your stomach is just empty and like just eager to have something expanding in, right? It's all constricted and lonely in there, right? It wants some company in the form of food. And so ghrelin, from your gastrointestinal tract, stomach in particular, you're releasing a bunch of ghrelin. Ghrelin is promoting AMPK in the hypothalamus. And then low blood sugar is doing it also. So if you're starving, if you have ghrelin, if you're doing your gummies or whatever, you're promoting AMPK in the hypothalamus. Now in the periphery, you see different effects turning on AMPK in the periphery. And there's a fascinating one in that over here, we see leptin inhibiting AMPK, hypothalamus, inhibiting AMPK. In the periphery, we see leptin turning on AMPK. It sounds counterintuitive until you think what leptin is. Leptin is released from fat cells as they're expanding. When fat cells are expanding, that means you've done a good bout of feeding. Your fat cells are expanding, your feeding is sufficient. We don't need to be hungry in the hypothalamus. Go ahead and burn stuff in the periphery. So if you activate AMPK in the periphery, you're increasing oxidation, fat oxidation. And if your fat cells are growing, that's a fine time to increase fat oxidation. And that's not the best time to continue eating when your fat cells are growing. So sometimes when you see things that look counterintuitive or don't look like they would make sense, if you think them through, they become a little bit more sensical. So summarizing AMPK, energy stress turns it off. Energy stress in the form of ATP relative to its peer group. So ATP hydrolysis, adenylate kinase, AMP accumulates, it gets activated. Once it's activated, it's inhibiting mTOR through several mechanisms, inhibiting mTOR. And it's promoting protein degradation. So there's tons of stuff is going to regulate it. You know, whether it's insulin, you see, you know, NAD, NADH, AMP to ATP, calcium, you know, PKB, there's LKB1, you know, there's leptin. All these different things have regulatory effects over AMPK. You don't need to know those, just the ones that we've been highlighting in class. AMPK has a ton of metabolic functions. The only ones you need to know are the ones you've been talking about, right? Oxidation of your macros. Let's break down proteins. Let's break down carbs. Let's break down fats. Inhibition of anabolism. Let's shut off glycogen synthesis. Let's shut off synthesis of your macros, right? So fatty acids. You're not gonna store fat. You're not gonna store carbs, glycogen synthase. You're not going to translate proteins. Let's shut off mTOR. And then also turning on autophagy in a lot of these. So what people associate fasting, the health benefits of fasting and cancer, stuff like that, of sort of recycling our cells. Now, mTOR summary, we have amino acids over here. We have protein. Okay, that's going through RAGs for mTOR. We have growth factors like IGF, insulin, a bunch of myokines, all of these sort of chemical activators, as well as mechanical loading. Growth factors, let's include mechanical loading over here. So let's turn on mTOR through Reb. You need both of these pathways to be active. You're not going to turn on mTOR by turning on a single one of these. You have to turn on both of them. Yeah, for amino acids, you have to get mTOR to the lysosome. Once it's at the lysosome, that's where it can interact with Reb. So we need to get Reb GTP loaded. We need to get RAG GTP loaded. Both of those need to be GTP loaded to get mTOR to turn on. So this two-step activation mechanism, that two-key system, it can't just be one, you have to have both. Because if you lose regulation of mTOR, that's death. And we're trying not to die in life. We're trying to prolong life. So the growth factor activation, so think, again, like IGF, secondary effects of testosterone, like insulin. Oh, I mean, just go through all of these, myokines that we went through. All of this, tuberin is breaking down the GTP into GDP. You need GTP-loaded Reb to bind to mTOR. And when it does, it kicks off, it displaces this inhibitory fat clamp, this inhibitory element. So the Reb binds to mTOR directly, turning it on, permitting its kinase activity to do its phosphorylating. Needs to be GTP loaded. Tuberin is breaking down that GTP. So that's Reb. Reb is the direct promoter of mTOR. Tuberin is the direct inhibitor of Reb's promotion. That's how growth factors turn it on. With amino acids, think of GATOR1 as the tuberin of amino acids. GATOR1 is the tuberin of the protein signaling, of the translocation of mTOR. RAG-A needs to be GTP loaded. If you're eating a bunch of amino acids, you're loading RAG-A with this GTP. GATOR1 is breaking that down, back to GDP, just like tuberin does to Reb. So in the end, we need both Reb and RAG to be GTP loaded. Directly upstream from that, we have tuberin and GATOR1 breaking down those GTPs. When they don't have GTP for RAG, mTOR dissociates. It's gone. It's not at the work site. For Reb, if it doesn't have its GTP, it's not binding to mTOR. It can't turn it on. So mTOR is off. Now, mTOR complex 2, complex 1. Remember, downstream from complex 2 is PKB, AKT or PKB. That's shutting off tuberin. That's inhibiting tuberin so that Reb can be activated. So complex 2 is unable to translocate. It's unable to relocalize mTOR to the lysosomal surface. Now, there's a lot of ways to draw a huge picture. This is from the literature. And this is what you're going to do on Wednesday. So on Monday, we'll finish our applications. So we'll just do a couple of applications today, and then we'll do the deep dive into applications on Monday. Not talking about AMPK at all. It's just going to be beginning to end applications. And on a Wednesday, you're going to draw mTOR. And there's some stuff on here that we've never talked about and you don't need to know. And there's other stuff that's not on here that you do know. But just a look at, here's complex 1, there's complex 2. What a kinase and a phosphatase and what transcription factor, remember a Cas space, enzymes. And so just this key of what this stuff looks like. And then what these lines mean, if it's a direct arrow, it's direct stimulatory modification. If it's a line with a little stop at the end, direct inhibitory modification. So those are good diagrams. There's a PKB diagram that's looking at the activity of PKB, both downstream and upstream. This is a former student's diagram. She did it from memory. I watched her do it. It took her about 10 minutes. She did in this room. I think she was sitting where you are. And it took her about 10 minutes and she did this out of her head. And no sources or anything like that. There's stuff, but at the end of it, I look at it and I'm like, oh, did you forget? Like, oh shit, I forgot. And so it is incomplete. But anytime you do these big diagrams, you're not gonna remember everything. Every single thing you know is not gonna come out in a diagram. 10 minutes later, like, oh, I totally forgot about X, Y, Z, LMNOP. There's a bunch of stuff. But this is a good one. This is a really good one. And so that's the idea of what you'll be doing on Wednesday as a team is with a whiteboard, get your team together and get as much mTOR drawn as you possibly can. As much inhibition, as much promotion, as much upstream, as much downstream, as much interaction, as much mTOR. Remember, mTOR is a synecdoche. Not the mTOR, and draw as much mTOR as you can. mTOR, the kinase. Draw the other stuff, draw signaling cascades. Now, sometimes people wonder, why do I need to know all of this cell signaling? Why do I need to know biochemistry, physiology? Why do I need to know how a cell works? Why can't I just have prescriptive sentences? Why can't I just memorize lines to tell people what to do? I'm going to be working with athletes, let's say. I'm going to be a strength coach, or I'm going to be a personal trainer, or I'm going to be physical therapy, working with people trying to recuperate bodies. Why can't I just memorize advice? And the reason is people who memorize advice are bad at their jobs, truly bad at their jobs. This isn't Courtney's opinion. This is fact. A memorized line only applies to a particular circumstance, a particular patient, a particular client, a particular athlete in a particular circumstance. Usually the average patient or the average client or the average athlete in the average circumstance. Guess what doesn't exist in life? The average patient in the average circumstance, or the average client or athlete in the average circumstance. That doesn't exist, which means all of these memorized lines about how to exercise, about how to eat, about how to behave in life, whatever your daily exercise and nutrition and all of this, common advice is most commonly wrong. So if that's all you have to offer people, you are literally a fraud. And I see this so often in athletics. I see this so often in medicine, whether it's physical therapy, doctors. It's populated, this isn't my opinion, it is populated by frauds who don't know how a body works. You need to learn this stuff if you want to offer somebody advice that is not a threat to the public health. You need to know science. And people so commonly ignore science and leap to prescriptions without knowing why they're prescribing things. The most important thing I can teach you is the science. The prescriptions, the applications, that's secondary. No matter who comes to you, somebody could come to you and say, I want to exercise. I need a nutrition plan, whatever it is. That person, maybe they're at risk of cancer, and what you tell them is lethal. Maybe they have diabetes. Maybe they are pre-diabetic in their blood sugar. Maybe they are on particular supplements. Maybe there's a family history of something. Maybe there's an exercise history that you have to contend with. If you give them the common advice, you're stealing their money and potentially harming them. I say this with our coaches here. I watch what our athletes do. It's commonly dangerous and uncommonly helpful because people don't know science. It is so much more important to understand how a cell behaves than to have a sentence about prescription. Okay, public service announcement over. Let's get into applications. So that's just to say, don't memorize applications, memorize science. Applications should be obvious if you know how biology works. So the two we'll do today are general health, which I'll spend very little time on, and then we'll get into eccentric loading, which people would say eccentric, although no dictionary in the English language permits that pronunciation, but eccentric or eccentric loading. You can say either way. So the general health lifespan is connected to AMPK positively. Lifespan is connected to mTOR negatively. Now, a lot of these studies are on creatures other than people. There's no reason to believe that people can't be counted among the creatures for whom lifespan is directly connected to anabolic cellular signaling. So in pretty much every study they've ever done, they're looking at exaggerated mTOR signaling is shortening a lifespan. And lots of studies talk about some of these mechanisms, but mTOR complex 2 does have some lifespan augmenting effects, but also through PKB, FOXO, inhibition of FOXO through PKB, inhibiting lifespan. So everything over here, what would be, autophagy would be enhancing lifespan, but mTOR is blocking ALK1, which would be promoting autophagy. So there's a lot of different mechanisms, different pathways through which mTOR complex 1 is detrimental to making it into your, 100th year of life. Application number 2 for general health, think blood pressure. Long-term regulation over blood pressure is the renin-angiotensin-aldosterone system. This is long-term regulation. You have angiotensinogen is released by the liver, and in the presence of low blood pressure, the kidneys secrete renin. And you convert to angiotensin 1. ACE, angiotensin converting enzyme, converts angiotensin 1 into angiotensin 2. Angiotensin 2 is vasoconstrictive and activates aldosterone, which absorbs fluid. You're going to reabsorb hydration. Blood pressure is thus increased. This gets dysregulated in life. Blood pressure so commonly gets elevated, and elevated blood pressure is a major risk factor, as you know, for adverse cardiovascular events. AMPK is a massive regulator of this blood pressure system, of the renin-angiotensin-aldosterone system, in a positive, healthful way. And we see a similar thing with vascular disease and ventricular hypertrophy, left ventricular hypertrophy. In elevated blood pressure, that pressure is pushing back into the heart. It has to beat harder. The heart has to beat harder and harder to eject blood if the pressure pushing back is higher and higher. So we're getting both heart health, vascular health, blood pressure in general. In the presence of elevated mTOR signaling, that's bad. In the presence of elevated AMPK, that's good, right? For lifespan and the kidneys. And I think glucose, diabetes, the percentage of adults with diabetes is scary these days. It's so high. I mean, fast forward the world 10 years, and like, what are the major threats to health going to be? I don't know. Climate change and diabetes; those are two good candidates, right? Based on what we're doing. And so you know how AMPK, what it is going to do to glucose metabolism, right? So ATP, right? ATPase, ADP, adenylyl kinase. We have AMP. AMP binds to AMPK, permits activation by LKB1, and AMPK is going to facilitate GLUT4 translocation, get carbs into the cell. It's going to turn on hexokinase, turn on phosphofructokinase, turn off glycogen synthase. AMPK is, if you have diabetes, you don't want insulin, you want AMPK. That's way better than insulin. With insulin, right, you're gonna get the PI3K, PKB signaling, and that effect, it's way worse. You're going to store all those carbs, you're gonna get insulin sensitivity problems, and chronically, glucose regulation is made worse by insulin, whereas AMPK, it is made better. But they're both going to mobilize GLUT4, but one of these is going to result in chronic impairment of glucose regulation. The other one is going to result in chronic improvement of glucose regulation. AMPK is much better for general health. Now, healing, let's say you get injured. A lot of these injuries are like, let's cut the skin and see how fast it heals. Healing is really similar in terms of the stages of healing. So, but look at how AMPK relates to healing. And early on, right, ICAR insulin, triggering different cascades. Insulin, PI3K, PKB, ICAR going through AMPK. You're getting increased vascular endothelial growth factor expression and more angiogenesis. So early on in healing, both mTOR PKB signaling and AMPK signaling are going to increase the amount of vascularization, the amount of nutrients being transported to the tissue. And you need those nutrients being transported to the tissue for healing to begin. And so you can see, again, this is like cut the skin. And this is like this DB. These are chronically, genetically hyperglycemic mice. And so here's the control and the percentage of wound closure. There's the control. The diabetic mouse with metformin heals better than the diabetic mouse without metformin. See the same thing over here, right? So capillary density, and there's a diabetic mouse, there's a diabetic mouse with metformin. Now, this isn't on people, and it's not on control mice given metformin, but when you see additional AMPK signaling early on in healing, you see improved blood supply, improved delivery of oxygen and nutrients and all that stuff required for healing. Now, long-term healing, AMPK compromises. Short-term healing, acute, brief, immediate healing, AMPK promotes. Long-term healing, AMPK is going to inhibit, because you're going to need proteins being translated. You're going to need proteins being deposited in that tissue. And so there's, again, like the cell death with metformin, right? So you're going to abolish translation in terms of hypertrophic signaling, and you're going to promote cell death. Not helpful for healing chronically. In the short term, it's very good. In the long term, you're getting apoptosis, you're getting problems. All right, let's end there, and then we will begin here on Monday. We'll begin with eccentric stresses, and we'll just do pure growth advice. From how to load tissue to the timing of balance, aerobic and anaerobic interactions, how long translation takes place, when to time things, supplements to take, all of that stuff, nutrition, all of that stuff we'll do on Monday. And then on Wednesday, it's your turn to draw out mTOR. And that's it, Core 2 day.